So Mr. Murra, if we can make 6 breakout rooms.
I think Durga Rao is here, right? Robin, whom did you talk to from NPTEL?
Yeah, I think Durga Rao is the one who's right now making the co-host.
Tell me, ma'am.
Yes. Yeah, we need to make 6 breakout rooms.
Ma'am?
We need to make 6 breakout rooms.
Okay, ma'am. Okay, ma'am. Okay, ma'am.
Yeah, yeah.
Oh, I think it looks like I can do that.
Oh, great.
Yeah, so we'll assign automatically. So before we get that, before we go there, I want to share this link. So I'll also just share my screen so you can see what I'm talking about. So hopefully what you all should see when you click that link is this. Yeah. And what we are going to do is just simply click this launch interactive, which will actually start a different, it'll open a new window, which you can't see, but I will. If I share my desktop, you'll be able to see it. Yeah. So it'll open a window like this. Yeah. So. And then it has, so this is a virtual lab. It's created by Howard Hughes Medical Center, HHMI. And this is really, it's a fascinating, exercise. It's a virtual lab. So what it does is that it's going to talk about lizards. And I mean, I know we are in an ornithology class, but it doesn't really matter. We are interested in looking at the concept. The exact traits that we work with will be different if it's a bird, but I think you will understand the whole process once you follow this through. What we need to do is just first we just listen to this 50-second story. Well, yeah, that's not much of a story, but essentially the idea is that there are different kinds of anoles, and what we are going to do is to try and understand what is called the ecomorph, which means that in terms of morphology, what organisms are more similar. But we are actually going to do, like, we are going to collect this data and assess this quantitatively. We are not just going to make a guess. Okay. And this is what we would do if we were working on something even if it's a bird or something else, this is how we would proceed. I know this is part of the module that looks at genetics, which actually comes in the second module here, which is phylogeny. But for the phylogeny to make sense, to put it in context, you have to be able to do the first module first. So you have to kind of bear with me while we do the module 1, and then we will move to the module 2. And the way this is going to work is that we are going to kind of have these breakout rooms, and there are TAs who know quite a bit of these modules, and you have to still work it out yourself. But despite that, if you run into any problem, then the TAs will be able to help you. So and there's a lot of materials to help you here, where there's a tutorial and so on. But for now, just go through the module 1, which lets you collect some data on these different types of anoles that are there. And once you have that, then we'll move to the next module, which looks at the genetic data from these from these lizards. And the question that we are asking is that if you saw that video, there were all kinds of anoles. So there are different forms of anoles. There are some that live on the tree, some that live on the ground, and some that live on in the canopy and so on. The question is, how did they evolve? And this is in the different islands in the Caribbean. So did they all evolve once and then they moved to these different islands, or did they evolve independently in each of these islands? Yeah. So we'll now kind of I'll stop sharing the screen. Yeah. So you only need to log in if you're going to come back and get your to restart at the same point. And if you have a Google Chrome or something like that, it will remember it based on that. But that's really up to you. So I'm going to create 6 breakout rooms, which will assign people automatically. Hopefully the TA should be spread around equally, and they will help you if you get stuck. But essentially you have to do the exercise yourself. They are just there to help you if you have doubts. At the end of the session, once you finish the module 1 and module 2, then what we are going to do is just have a small discussion among your group in your breakout room about what are the main things that you learned from this experience. Okay. It's just like 2-3 points, not but do discuss and then make those 3 points, and then we'll meet in the common room. And if everyone can kind of synthesize what they learned, that will be useful. All right. Any questions at this point?
So, I am using mobile.
It may not work very well. But you can give it a shot. I mean, I know that a lot of people have tried it out with mobile. You can give it a shot and see how it works.
Okay.
Excuse me, sir. I have just joined the class with another device, my laptop. I don't have the link which you just shared.
Oh, I see.
Can you please resend?
Yeah, Jobin, can you? Yeah, can you just resend? I think Jobin's going to send the link again. And once you're in the breakout room again, the TAs will copy and paste the link for you so that it's with you in the breakout room as well. So don't worry about that. So the idea is let's give us for module 1, maximum about 15 to 20 minutes. So you have to kind of rush through this a little bit. So try and be very quick. By around 3:30, 3:40, we should be done with module 1. Then we go to module 2. Yeah, and then max around 3:45 or so, we finish all your activities and then get back to the main room. Okay, so over to breakout rooms. Hi. So those of you who are still in the main room, if you have any questions, please tell me. Otherwise, you have to kind of get into the breakout rooms. Devika, can I request you to just assign people, unassign people as they come? If you can request them to, or you can assign them to some room.
Yes, absolutely.
Okay, thank you. So now I'll head over to these rooms one by one.
Sure. Prasad Kamath, are you here? Okay. Anu? Is Anu here or FK? Viraj, did you just join? Okay, I'm going to assign you, Viraj, to some breakout room. So everyone is in separate breakout rooms and they are doing some hands-on session there. So I'm just going to assign you to a breakout room. Let me see how I can do it. Not sure of how to do it actually. Durga Rao, do you know how we can put Viraj in a breakout room?
Ma'am,
Or so we need to put Viraj in room number 2, breakout room number 2. Do you know how we can do that? Viraj just joined. He joined late.
in room number 8,
We can put him in
you want
room number 2. But how do we do that?
your voice. Tell me, ma'am.
Um, can you put Viraj to room number 2? Durga Rao?
Okay, ma'am.
Yeah.
Okay, ma'am.
Yeah. Room number 2.
Okay, ma'am.
Okay, Viraj. I am not able to do this. I'm just going to Google it, how to do it. Punil Gajar, did you just join? You can unmute and talk actually. Aisha, so let me tell you that everyone is in different breakout rooms at the moment doing some hands-on work. So I'm just going to put you in one of the rooms.
Actually, ma'am, I'm Archana Gajar, and
Okay.
I could not see anything on the screen. Actually, earlier it was, sir was there, but now
Okay.
it is showing meeting topic and post.
Which room were you assigned to?
I think room number 3.
Oh, you were assigned to room number 3.
Yes.
Okay. Let me
I
see.
think I should copy the link. Or
Which link?
on my screen, it is, I can see the meeting topic, then host, password, invite link, and participant ID. So instead of the Zoom meeting,
Okay. No, I can't. So I can't put you. Can you just try joining room 3 again?
I don't know how to join room number 3.
Okay, because I can't assign you because that room has already been assigned to you.
Okay.
Just go to
Okay, I'm trying to join break.
Yes. Ahana and Beena, I think you've just joined, right? So everyone else is in different breakout rooms doing some hands-on sessions. I'm going to put you both in room number 4. Ahana and Beena.
Hello.
Hello.
I was already in group number 1. Can you please?
Oh.
Actually, I've joined from another device.
Okay, but I can't assign you to any other room now because a room has already been assigned to you.
Okay.
Just try joining in again in room number 1.
Okay.
Musamillu, I'm just going to put you in room number, let me see, 4. Deepak Sinha, you just joined or you just got out of a breakout room? Deepak Sinha, are you there? Okay, Mrinal, did you just join the meeting or did you just get out of a breakout room? Mrinal, are you here?
Yes, ma'am.
Yeah, did you just join or did you just come out of a breakout room?
No, I just joined, ma'am.
Okay, so everyone else is in different breakout rooms. I'm just gonna assign you to room number 2. Okay?
Okay. Thank you.
Krishnendu Das, did you just join or did you just come out of a breakout room? Please feel free to unmute yourself today and talk to me. Okay, Pushpa Suresh, did you just join the meeting? Pushpa Suresh, are you here?
Yes, ma'am.
Yeah, did you just join the meeting or did you just come out of a breakout room?
I just joined the meeting, ma'am.
Okay. So everyone else is in different breakout rooms doing some hands-on session.
Okay.
So I'm just gonna assign you to one of the rooms.
Okay.
Hope everyone here is on their laptop and not on your mobile because it will require you to do some hands-on work.
Oh,
So if you're on your mobile, please try and get on your laptop.
okay.
Adil A, are you here? Adil A, did you just come out of a breakout room or did you just join the meeting? Okay, someone called user has just probably joined the meeting. Can you please unmute yourself and talk to me? Um, can you please rename yourself? It says user at the moment. Okay, wildlife of DJ. I guess you've just joined the meeting, right? Or did you come out of a breakout room? You can unmute yourself today. Okay, I'm gonna just put you in one of the rooms. So user and wildlife of DJ, I think you've just joined the meeting, both of you. Everyone else is doing some hands-on session in different breakout rooms. So I'm gonna assign you to one. I hope you are on your laptop and not on your mobile because it needs some clicking of links to do. Deepak Sinha, are you here? I can't hear you at all. Hello? Rekha, I guess you just joined the meeting. Can you hear me?
Hi, Devika.
Hi, Robin.
So I was thinking that, if new people join at this point,
Mhm.
maybe don't send them into the room.
Yeah, makes sense. Right.
Yeah, because I just noticed that some people come in and then, like we have to start all over again. So it's a little disruptive.
Right. I guess it's okay. It's already like those who have joined in right now are already almost 40 minutes late. So it's okay.
Yeah. You can just give them the link, that is this link, and then they can do it themselves later.
Yeah, will do.
Okay, cool. I'll just head out to the breakout room again.
Sure. Well, Rekha, I guess you've just joined and you heard that. So I can't put you in any breakout room now, but Robin just shared a link on the chat. So you can just explore that link. Basically, that is what everyone else is doing in their rooms. Those who have just joined, Govind and Vishwanathan. Everyone else is doing some hands-on session and hands-on work in their respective breakout rooms. But if you can see the chat, there's a link there shared by Dr. Robin. You can just check that link out and explore. If any questions, please go ahead and ask me. Govind Girija. Actually, this session cannot be recorded because everyone else is doing some hands-on work like in the breakout rooms. So I'm not sure how much uploading the video is going to help, but I will share the link.
Okay, I will join then.
Yeah, I've just shared the link, which you can explore on your own.
Okay, thank you.
Yeah.
Please share the link again.
Yes, just shared it right now again.
Yeah, I will do it. I will take it. Do it. Try it later. Yeah.
Sorry?
I will try. I will do it later because I tried. I'm not able to do it.
Okay.
I've done the calculations and the video is, it is the continue tab lead me to your video. Is that all after that?
Sorry, was someone here talking to me?
After the measurements, it went to a measurements table.
Oh, is this about the link that has been shared?
Yeah, the breakout room. Once we started.
Okay. Sorry, I don't use that software, so I'm not aware of it. But probably someone among the TAs can help you. Can you just okay. Just give me a second, I'll figure something out.
Once the measurements have been done, I've been led to the table to fill the relative lengths. So after that, the tab led me to a video.
Yeah.
Is that it?
Oh, then you have to say, did you say launch? Just a minute, I'll just open it myself. Did you say launch interactive after the video?
I started with the link which went for launch interactive. Then I was given images of the lizard to group.
Okay.
Once I grouped them, then it went to the continue button, took me to the measurement.
Okay, Robin should be here in some time to help you with it.
Yeah. So please tell me, after you did the measurements, what happened?
After I did the measurements, it led me to a table where 3/4 of the table was auto-filled and the balance were left me to fill by calculating. So I did that and I went for continue button and it went to a video straight away.
Yeah. So the video is like a synthesis that it tells you, from the data that you've collected, what it can answer about your question. So you don't need to watch that video fully. You can switch back to module 2 after that.
Okay, right. Thank you.
Yeah.
Thanks, Robin.
Yeah, no problem. Essentially, what this is trying to do is that, initially this module asks, so let me just, since all of you are here, I'll just share my screen and show you also what this is about, what's happening. So we have a module which is like this. So initially there are all these anoles. This is the first module, ecomorphs, and it asks you to group the lizards that you see. So, and they don't say how you should group it. It's totally left to you. So you say, okay, this is green, and these two are green, so I'm going to keep them together. And then these are kind of brown, so I'll keep the brown ones together. And something is like slightly different color or something else, I mean, whatever you feel like. And then you have to kind of make a label, and you can say, oh, these are brown lizards, and I put the brown lizards here. Then I add another label which is maybe twigs, and then I do that, I put that here. Another is green lizards, so I put that here. Yeah. Yeah, somebody's watching the video. Yeah. Can you mute the video?
Yeah, I'm just trying to.
Yeah. So basically then after that, yeah, you have to start measuring these. So what you've done is you've looked at what it is for you, what it seems like the groups, but then you have to collect data on this, and for that they already have some data on, like they have X-rays. So if you click on any of those species, then it lets you collect data. You can say, yeah, this is the the body length by measuring this, and then this is the hind limb length, and so on. Yeah, and then this is the tail, and so on. I mean, you have to do it carefully. I'm just doing some random thing. Yeah, and once you record the measurement, which is not this, but I hope you get the idea that you can carefully collect all of this data. See, actually, this is how it should be. Yeah, something like that. So that is hind limb length, and then you have to collect the body length, which is this, and then you measure the tail length. You have to go. Yeah, sorry, I'm not doing this very carefully, but I hope you got the idea. Sorry, I didn't click save. Yeah, so similarly, you just have to do, I'm just trying to, yeah, you have to do multiple species. And then what happens is that you can look at the results table. So until I do all of those, it won't show me the results table, unfortunately. So I just have to do this, this click, click, click thing through all of them, record this measure, and then record that. Yeah. And there's one more remaining. Actually, there are two more remaining. So the first is the hind limb. So the point is that you're now measuring the X-rays of all of these animals, which gives you some data of them. So there are a few more actually. And if you have doubts, there's something called tutorial, which tells you where you have to measure from. Oh, sorry, it's supposed to include the head. I think I did not include the head when I was measuring the body. So essentially, I did the hind limb right, almost. But the body length is actually from the tip of the head.
Robin?
Yeah.
Yeah, so my the exercise is done with my group. So should we all get to the main room now?
Yeah, please.
Yeah, okay. Yeah, I'll just inform them.
So, yeah, unfortunately, this is a 15-minute exercise, and I'm trying to get it done in a few minutes. So it's a little bit Yeah, I think we are mostly done. And then there is this thing called toe pad. Yeah. So what this does is you have to measure the lamellae, on the toes. And again, if you're not sure how to do, here's how you do it. So you have to click on each of these, and it gives you a count of how many there are. Yeah. So technically, you're supposed to kind of, click on each one. Yeah, terrible. Yeah. So basically what it's telling me is that I did it badly. It's a little bit easier. No. Yeah. And then once you have done this, you essentially get a table. And this table summarizes your measurements and then the reference measurements. So this is what it's supposed to be like. And yeah, of course, I made a lot of mistakes. But that's okay. And then you have to kind of calculate this relative hind limb length, and you have to put that number here. I hope it won't let me, but we just move on. Yeah. And then there's a 2-minute video to watch, which you don't have to watch right now. You can watch it later, but please do watch it. But essentially what it tells you is that the grass bush anoles have long tails, and there are these different ecomorphs that are associated with different morphological features. Yeah. And then you can say, oh, now do you want to regroup them? And you don't have to do the quiz for now, but it will give you the graph. Okay. And this graph has relative hind limb length to relative tail length. And what it tells you is that these are these ecomorphs. Yeah. So this is actually not the exercise that we want to do. What we want to do is module 2, which is, I think, here. And what this module does is that you're asking, yeah, maybe I'll just let everybody join in, and then we can finish this up. So all the rooms should have closed now. Is that right?
Yeah, I think.
I think there are more people in the room as well right now. Even I can see 7, 8 somewhere.
But I closed all the rooms.
Yeah. Okay, yeah. I think they're closed. Sorry.
Yeah.
Yeah.
We just to be sure, I'll just delete all the rooms. Yeah. So now no rooms are open. Okay. So I hope I realized that this may have taken a little bit longer. Maybe you can continue this exercise later as people are suggesting. But I just want to share my screen and just take you through what what we really wanted to get to was this, which is this module 2. And I'll just play this video. I think it gives a good idea.
On each of the Caribbean's 4 largest islands, Puerto Rico, Jamaica, Cuba, and Hispaniola, we find the same distribution of similar-looking lizards. Each island has slender grass bush anoles with long tails, long-legged trunk ground anoles, short-legged twig anoles, and canopy anoles with large toe pads. Did each body type evolve once and then spread to the other islands? Or did each type evolve independently on each island?
Yeah, so I know that some of you may be a little bit disappointed that this is that this has lizards, not birds, and we are in an ornithology class. But you can imagine any other bird. You could be thinking about Darwin's finches or any of those. The idea is the same. Darwin's finches also have different beak shapes, and you could ask the same question: did they evolve once, or did they evolve multiple times? The question is the same, just the organism that with which we are studying them and the traits that we are studying change and are different a little bit. Yeah. So don't worry about the specific organism or that you don't know how lamellae matter for lizards, but it's not something that you would measure for birds. Yeah. Can you hear me now? Can somebody else indicate if they can hear me?
Yes, we can.
Yes, sir. You're clear.
Okay.
Yes.
Thank you. So, so the idea has been that we have these ecomorphs, and we are trying to understand how they evolved, and those are the kinds of questions that scientists ask frequently. So now I was just wondering if we can have a few volunteers to summarize what they learned from different groups. So if group 1, if anybody wants to say something.
I mean, which is the group 1, group 2?
Okay. I thought everybody knows.
I was in group 1. I think
Yeah.
there are people from my group here. Yeah.
May I, sir?
Yeah, please.
Sir, am I audible, sir?
Yes, you are.
So firstly, sir, I must thank the faculty for such a fantastic module been presented to us. As far as I'm concerned, in person, it really actually don't matter to me that I was working on lizard, though I'm into ornithology.
Sure,
because this is a fantastic example wherein 4 lizard are on an island, and each of them having a different habitat, which speaks in parallel to the avian also, where the course went on, where the birds carve their own niche in their own habitat. So maybe because of competition and
Yeah.
for various things. So I won't take much of time, sir. Thank you. This is
Yeah.
something which I could draw in one line.
Yeah.
that this was very interactive and very beautiful way to present it. Thank you,
Sure.
sir, and thank you, team, sir.
Yeah. Thank you. So my question now, maybe the second team could say, what what were the three main things that you looked at, in this module, and how does it affect your thinking about species? Actually, let's not do it by teams. If anybody wants to, um, uh, just
I just understood that the habitat division is based on the tail, which is long or short, leg, which is long or short, and if the lizard is having a number of larger toe pads, they were higher up in the trees, and in each floor they were having different evolutionary
excellent
adaptations in there.
Yeah, excellent, excellent. So that's what we can tell by quantitatively looking at these characters, right? I mean, first you just looked at the animals. So this is the equivalent of bird watching, where you just see the bird, and then you're trying to kind of get some ideas, and then you go to the next step, which is you collect a little bit of data, and that is your ecomorphs, which tells you how they are kind of adapted. And then there is the next level of data, which is the genetic data. So can somebody tell me what did you learn from the genetic data? Maybe a group that finished. Yeah, please, Radhika.
Yeah, so I think the phylogeny data kind of indicated that evolution can happen not on the the genetic kind of tree, but also on the habitat. So I think that determined how the species could evolve independently, like the convergent evolution concept that was explained.
Nice, nice. Yeah. Anybody else has anything else to add to this?
Yes, this is an example of radiation, like Darwin's finches, what was shown in the module. And I thought it was very interesting that when we measured the hind limbs, and afterwards when they were compared, the hind limb ratio was taken with the body. So
That's
I thought that was very interesting.
Yeah. I know what you're talking about. And let me just, maybe I can just share my screen and show that. I'm not sure if everybody got to this, but in the ecomorphs module, at the very end, there is this graph that people reach to. And this is relative hind limb length and relative tail length. And these are the ecomorphs that people are talking about. Okay, if those of you who didn't manage to reach this phase, so there are some that have relatively larger hind limb and large tail. So if you have long tail and long limbs, this is where you are. And then some are really squat and short. Yeah. Yeah, thank you, Mukta, I think, for that summary. Anybody else wants to add something to that?
I want to add a point related to phylogeny, sir.
Please.
It's quite interesting, the exercise, and thank you to the TAs and like very good lecture. Actually, I just come across your lecture also related to ornithology. It's so nice, sir. Actually, through phylogeny, we all of us we come to know the common ancestor and what way the grouping. That was too fantastic. But basically, I have a question in my mind. Though we have a conventional mode of classifying or taxonomically classifying organism, it's not only birds or animals, whatever we are studying, we have a very good tool nowadays, molecular based one. In that case, should we do experimental analysis in both the way, conventional as well as molecular based, or can we restrict ourselves to molecular basis alone? This is what the question I had in my mind. Will you please clarify, sir?
Yeah, it's a good question. Yeah. And I think some of it is philosophical, the thing that we are always trying to do is trying to understand the truth. And long back, what people thought the truth was by measuring. You collect specimens and you look at the morphology and you think that this is what it is. And today the idea of truth has changed a little bit. And maybe a decade back, people thought that once you have genetic data, they think that that is the end of it. Like, yeah, you have genetic data, so this must be the truth. And then slowly people start realizing that it's not just genetic data, but the number of genes also matters. So it's not enough that you have one gene, because that gene probably tells you a different story. So you want more genes. So then people started saying you need like 6 genes or 10 genes or something like that. But now you're looking at like whole genome level data. So people are saying, no, that's not enough. You have to look at the entire genome. So what is truth is it depends on the times that you live in, the kind of data that you collect. But as of now, what it looks like is that people say that genetic data by itself is not enough. You need to have an integrative approach. So you will find more recent bird taxonomy papers which will have some amount of genetic data, some amount of bird song data, acoustic data, that is, it can be calls or songs. They may have morphology data, they mostly always do. And there is an integrative approach that is taken to that. People also add other ecological traits to this, depending on what species it is. So I think taxonomy is kind of, is because it's an old field, they're a bit hesitant to accept change, but the truth of the day is changing. So we need to adapt and include all possible evidences, I would say.
Yes, sir. Thank you, sir.
Yeah.
So one more clarification, sir. Is there any way cloning approach was employed in ornithology field, sir?
I don't think, I'm not sure. I don't know. There we have a lot of TAs here. Does anybody know of any cloning? Anand Krishnan is my go-to person for bird trivia. I'm not aware of too much of the cloning work in birds. In mammals, quite a lot has been done. There's a whole lot that's done with pets. In China, there are companies that will clone your dog for you and so on. But with birds, I haven't seen this too much, but I have not followed the literature as much either. So, yeah. But cloning does not give you too much information about evolution, in that sense. Yeah, you just create another copy of the same individual.
Yes. Yeah, thank you. Just for the curiosity's sake. Yeah.
Sure. Yeah. Sorry, Badri has his hand up. Can I ask him to go first? Yeah, please.
I'm sorry, I could not complete even the first step of the exercise.
Sure.
But the initial part of the exercise was to measure the lizards. And I'm not sure whether there was any information on the habitat which each one of those 8 lizards preferred. Shouldn't the way one be going over it be first to see the lizard, in what sort of habitat does it live, and what sort of tools does it need to live in that habitat, and then does it have those particular tools, instead of rather jumping into a measurement first of all?
Yeah, no, I think I understand what you're saying, and that's very true. I think that's what an ecological research would normally, that would be the way that they go about it. In this case, there was a point that they're trying to make, which is that you can infer the habitat just by looking at their morphology.
Okay.
So let me give you a bird example. So, tarsus length is a very good indicator. So birds with long tarsi are often not perching birds. They are ground dwelling birds. Whether you take, pretty much
pipits and
owl or even pipits, or, but as you go more canopy dwelling, more perching birds, they have shorter tarsi, right?
Okay.
And this is similar for the wing length also. The more pointed wings birds are the ones that are mostly on the air for long periods of time. But rounded wings and those birds don't move as much. So I think what this exercise was trying to do was to tell you that if you have just the morphology information, you can infer the ecology of the species. So you're right that you can go, ideally you should go from ecology and then do morphology, but the association is so strong in some cases that you can, infer one thing from the other.
Okay. Thank you.
Thank you, Badri. That's a very good observation. Yeah, so can I have Sarkar and then Krishnendu?
Good evening once again, sir. So I'll request if you can be a bit more elaborate on your work on the Solakili and rather the laughing thrush, where it actually came into a different genus, Montesecla.
Yeah.
Sir, what was the finding actually? Can you be a bit elaborate? Because in my personal capacity, I was not a bit satisfied with that lecture part. I could not make out what brought them into a different genus, sir. If you be kind enough, sir.
Yeah.
Thank you, sir.
I can also share a link of a talk that I gave that talks more in detail about this. Essentially, what we understand today about these birds is that they probably came in from somewhere in the Himalayas, and with as the climate changed, they moved up the mountains and they got isolated. And they are tied to this habitats which are found on mountaintops. So when they move, when the habitat moves, they're able to move down the mountain. Otherwise, they don't. They are stuck on the tops. And in fact, the Sholakili and the laughing thrush seem to be stuck across one particular barrier for over 5 million years. They've not crossed that 40-kilometer gap. And that's why they are different species. So the use of molecular tools, which was the lecture that this was under. So we had 2 species earlier of laughing thrushes, but because we have this molecular tools, we are able to kind of look at these differences between the populations, and we know that they're actually supposed to be 7 species. So I think that's a quick summary.
Thank you, sir. Thank you, sir. Thank you, sir.
Krishnendu, you had a question, you can go.
Yes, I wanted to ask, my question will be rather a straightforward question. You mentioned 3 things in the synthetic approach. You take genetic data, you take acoustics into consideration, and you also take the morphology part. So my question is, in one of the earlier lectures in the course, there was an example discussed about greenish warblers, which was thought to be a single species, but later on detailed studies on acoustics and on genetic analysis found it to be 6 different species. So my question is, can 2 species have more or less similar genetic data, but still differ by their bird songs and maybe, yeah, is it possible?
Yeah, so see, the thing is that, so first I'll give you a bigger picture answer. The question goes back to what is a species? And, we are trying to kind of group birds into these different groups. Birds, whether they follow the rules that we are following to group them, that is the question that needs to be asked. And the way people do that is by doing playback experiments, see if they respond to each other, and so on. I mean, the older ideas were to kind of actually put them together physically and see if they breed, but that's not considered necessary anymore. I would say that there can be isolation between species, but there are some thoughts that say that you have to be at least 2 million year old divergent for to be called a good species. Okay, so but this is also now considered arbitrary, because there is something called speciation with gene flow, where some amount of gene flow is possible, and still you may find speciation which is keeping populations apart in somewhat the kinds of ideas that you talked about, where maybe other traits are different. So ecologically and acoustically they're divergent. So at least theoretically there are some ideas that have kind of pushed that forward. So yeah, this is something that is ideas of active research today.
Thank you.
Yeah. There's a lot of questions. So maybe I should go to the Google form.
And one thing I just wanted to mention, the reading materials that is being provided, sometimes those papers are from journals which are not accessible to us. I mean, we cannot access this. So is it possible to provide the link of the article directly?
So I think that, yeah, so we'll have to try out we have to try and figure some alternative for that. I have at least made sure that we have at least the articles are available through ResearchGate. Yeah, I mean, there's a legality complication about this, but I think if those who are interested in the paper can write to Devika